Have you ever wondered if we could get a truly accurate picture of what someone eats without having to just ask them? What if we could actually read the body's chemical signature to figure out the real connection between diet, our gut microbes, and our health? Well, that's exactly what metabolomics lets us do. So here's how we're going to break it down. We'll kick things off with the big problem: how do you even measure diet accurately? From there, we'll get into the core concepts of metabolomics itself. Then we'll look at the super important link between our gut and metabolism, check out a really interesting case study on something called metabotypes, and then, of course, we'll talk about common pitfalls to watch out for before we wrap it all up. Okay, so before we can see why metabolomics is so useful, we have to get a handle on the problem it's trying to solve. And honestly, the first big hurdle in nutrition science is just figuring out what people are actually eating. It sounds so simple, right? But it's a huge challenge. You see, on one side, you've got the classic self-reported food diaries, and they're just notoriously unreliable. I mean, people forget what they ate, they're not great at judging portion sizes, and we know for a fact there's a lot of systematic underreporting going on. But then on the other side, you have metabolomics. And what that gives us are objective biomarkers. These are like chemical fingerprints in the body that give us direct, independent proof of what someone's been eating. And these markers can be incredibly specific. For instance, if you find a molecule called trans-4-hydroxyproline, that's a dead giveaway for red meat consumption. Or if you spot ergothioneine, that points right to someone eating mushrooms. These aren't just guesses. We're talking about chemical facts here. All right, so we know these chemical fingerprints exist, but how do we actually find them? Well, that leads us right into the core concepts of metabolomics and how this whole science works. So what is metabolomics? It's the science of measuring and quantifying all the small molecules, the metabolites, in a sample. Think of it as a really powerful phenotyping tool. It's measuring the end products of all our biochemical pathways, which, as you know, are shaped by both our genes and our environment, including, of course, our diet. Now, to make these measurements, scientists usually turn to two main platforms. First, there's mass spectrometry, or MS. This separates molecules based on their mass to charge ratio, which makes it super sensitive, perfect for finding molecules that are present in really tiny amounts. Then you have nuclear magnetic resonance, or NMR. This technique excites the nuclei of atoms with radio waves, and its strength is providing highly accurate quantification, even if it's not quite as sensitive. Each one has its job, and the one you choose really depends on the question you're asking. And within those platforms, you've got two main strategies. The first is untargeted metabolomics. This is like casting a huge net to see everything you can catch. It gives you this global profile of all the molecules you can possibly find. It's really a discovery tool. On the flip side, you have targeted metabolomics. This is more like a sniper rifle. It's designed to precisely measure a specific, predefined set of known metabolites, and it does it with extremely high accuracy. Okay, this leads us to a really fascinating part of our metabolism. We aren't the only ones producing all these metabolites. Turns out, a huge player in our metabolic story is the trillions and trillions of microbes living inside our gut. And just to give you an idea of how much influence they have, at least 15% of all the metabolites circulating in our blood don't come from our own cells. Nope, they're actually derived from the activity of our gut microbes. It's like having a whole chemical factory running inside of us. These molecules that the microbes produce, they have a name: xenometabolites. That xeno part means foreign or non-self, and they're made when our gut bacteria get to work metabolizing things from our food, or even stuff our own body makes, like bile acids. Let me give you a perfect example of this in action. Say you eat foods like walnuts or pomegranates. They have these compounds called L-gitannins. Now, our own bodies can't do much with them, but our gut microbes, they can. They take those L-gitannins and metabolize them into these really interesting bioactive metabolites called urolithins, and those our body can absorb and use. See, it's a crystal clear pathway from diet through the microbes to us, the host. But here's the thing, this whole process of making urolithins, it's not the same for everybody. And this variation leads to a really powerful case study in what scientists call metabolotypes. So depending on which specific urolithins a person's gut microbes produce, we can actually classify them into one of three distinct urolithin metabiotes. You've got metabiota A and metabiota B. They're both producers. They just make different kinds of urolithins. And then there's metabiota 0, and these are the non-producers. And why does this matter? Well, these differences have a real impact on our health. Research has linked metabiotype A with a more protective health profile. But metabiotype B, on the other hand, has been associated with an increased risk for cardiometabolic issues. So yeah, your microbial signature has real-world health implications. But here's the really hopeful part. These profiles aren't permanent. They're not set in stone. Your xenometabolite signature is actually dynamic. For example, the levels of other helpful xenometabolites like indole-3 propionic acid, which has great anti-inflammatory properties, have been shown to change when people make lifestyle changes like exercising or losing weight. Now, just like with any powerful technology, you've got to be aware of the common pitfalls. It's so important to know how to interpret the data the right way. One thing that happens a lot in these untargeted studies is that you find unknown metabolites. These are basically signals that don't match any known molecule in the databases we have. And you might think that's a failure, but it's not at all. In fact, every single one of those unknowns is a potential door to a brand new scientific discovery. Another really key point is that your choice of analytical platform is absolutely critical. We talked about this a bit earlier, but it's worth repeating. If you're on the hunt for molecules in tiny amounts, like a lot of these xenometabolites, you need the high sensitivity of mass spectrometry. But if you're looking at high abundance molecules or getting the exact quantity is the most important thing, then NMR is going to be your better bet. And this is maybe the most important rule in all of science, and it definitely applies here. Correlation does not prove causation. Look, finding a link between a metabolite and a disease is an exciting first step for sure, but it always requires more work, more investigation to prove that the metabolite is actually causing the health outcome. All right, let's pull all of this together with a quick summary and a couple of questions just to make sure we're all on the same page. So, to recount the big points. 1. Metabolomics gives us an objective way to measure what people eat, getting us away from those flawed self-reports. 2. Our gut microbiome is a major player, creating all these bioactive xenometabolites from the food we eat. And 3. Concepts like the urolith and metabotypes show us a direct link between our diet, our microbes, and our personal health risks. So, I'll leave you with a few things to think about. First, why do we consider that untargeted approach a discovery tool? Second, what's the main source of those circulating urolithins we talked about? And finally, after you find a correlation in a study, what's that all-important next step? The answers to these questions really get to the core of how this science works, and, more importantly, how we can use it to get a much clearer picture of the incredible relationship between our diet, our microbes, and our health. What do you think the answers are? Leave your answers in the comments, and if you like this video, hit that like button, and please consider subscribing to my channel.